位医药界同仁，大家晚上好，我是上海反目股技术支持王玉喜，非常感谢大家参加此次采用 PBPK 模型预测首次人体 PK 的线上公开课。通过临床前种属的药代动力学推算人体 PK 是准确计算首次人体剂量的重要基础。近年来，各大制药企业在临床前研究阶段已将生理药代动力学 PBPK 模型作为预测首次人体 PK 的常规方法。本次公开课将重点讨论当前工业界采用 PBPK 模型开展首次人体 PK 预测的策略，包括该方法在口服药物首次人体 PK 预测时的重要影响因素，比如影响吸收、分布、代谢的因素，以及对应的不确定性分析。最后将通过案例讲解如何通过 PBPK 模型减少不必要的临床实验，以及如何预测人体的有效剂量。今天我们很荣幸地邀请到美国 Simulations Plus 公司模拟科学副总裁 Neil Miller 作为本次公开课的主讲人。Neil Miller 由2020年加入 Simulations Plus， 主管 PBPK 模型在制药领域的高级应用，并引导 PBPK 模型的优化。加入 Simulations Plus 公司之前， Neil Miller 在 GSK 工作了24年，担任系统建模与转化生物学部总监，领导一支由 PBPK 和 PKPD 资深专家组成的团队，承担的项目包括预测候选化合物的人体有效剂量，采用基于生理的生物药剂学模型指导药品的开发等。Neil Miller 的讲解过程中，大家如有问题可以随时提问。在讲解结束的问答环节，我们将会对大家提出的问题做出解答。讲解结后讲答环节将分为2个部分。第一部分根据报名时预留的问题，筛选出了部分问题，先进行答疑。这部分问题我们也提供了中英文字幕。第二部分是针对今天直播间中提出的问题进行解答。参会人员可以用中英文的形式在直播间里留言，我们将遴选部分问题，有请 Neil 进行解答。接下来，请 Neil Miller 开始今天晚上的报告。
Hello, everyone. My name is Neil Miller, Vice President for Simulation Sciences at Simulations Plus. Firstly, I would like to thank the organisers for giving me the opportunity to talk to you today about first-in-human physiologically based pharmacokinetic predictions. The take-home messages from today's presentation are that I will give you an overview of what PBPK is and how it is used. I will also cover in more detail the application of PBPK for first-in-human predictions. And then finally, we will look at some first-in-human PBPK case studies. So what is PBPK? The standard definition of PBPK is physiologically based pharmacokinetic modeling. And this is a mathematical modeling technique for predicting the absorption, distribution, metabolism, and excretion, the ADME, of compounds in humans and animals using physiology and compound-specific physicochemical data. An alternative definition that I use is PBPK is a platform for bridging people and knowledge. That is, quality PBPK models are built when discipline experts data and knowledge are integrated together. That is, the chemists, the biologists, the DMPK scientists, and the biopharmaceutics scientists in clinical pharmacokinetics work together to build a quality model. There is a saying that a picture paints 1,000 words. And so if we were to ask a DMPK scientist, what is PBPK? These are the pictures that would come into their mind. The focus would be on systemic disposition, and the PBPK model would be a whole body PBPK model where each compartment represents a tissue in the body. And each of those tissues would be split up into various compartments. So we would have the vascular space, the extracellular space, and the intracellular space. If we were to ask a biopharmaceutics scientist, what is PBPK? The picture or the diagram that would come into their mind is that of the advanced compartmental absorption and transit model, which is the diagram here on the left, where the GI tract is split up into 9 compartments from the stomach down to the ascending colon. And each of those compartments, there are many processes occurring simultaneously. So we have transit in to the compartment from the previous compartment. This could be dissolved or undissolved or unreleased material. Within that compartment, there is dissolution, precipitation, degradation, and then transit out of that compartment. There is also transfer or absorption of the compound into the enterocytes and also into the systemic circulation. So depending on your scientific discipline, you may think of different PBPK models, different diagrams, but there are common features between those models and diagrams. So what is PBPK to everyone? Well, PBPK is a model that contains a system, a physiological structural model. That model has mechanisms and drug-specific parameters. So PBPK to everyone is a modeling technique. PBPK is striving to be mechanistic. PBPK is attempting to separate physiology from the drug-related parameters. PBPK all routes of administration, and PBPK is increasingly accepted by the regulatory agencies. So we've looked at what PBPK is. Now let's look at what PBPK is not. PBPK is not a single model. PBPK is not just for intravenous and oral dosing. PBPK is not simply an alternative to allometry. And PBPK is not just for small molecules. Next, I would like to focus on the question of how is PBPK used? To answer this question, we will use a publication from Zhang et al. In the Journal of Clinical Pharmacology in 2020. The title of the publication is Application of PBPK Modeling and Simulation for Regulatory Decision-Making Impact on US Prescribing Information. And this was an update on the 2018 to 2019 submissions to the US FDA's Office of Clinical Pharmacology. You will see from the diagram on the right-hand side that 40% of the applications focused on drug-drug interactions and enzymes, and 16% focused on drug-drug interactions with transporters. But if we focus on the 6%, that includes applications of first-in-human PK predictions. And there are various other applications such as pediatric modeling, etc. So there are many applications for PBPK. It is used in many situations. So PBPK is used for first-in-human PK predictions. And here we see a number of publications that highlight that PBPK works for this application. So on the left-hand side, we have a publication which focuses on the simulation of human intravenous and oral pharmacokinetics of 21 diverse compounds using physiologically based pharmacokinetic modeling. And this was work carried out by Pfizer. And their conclusion was that the simulation results using PBPK were shown to be superior to those obtained via traditional one-compartment analyses, and in many cases, this difference was statistically significant. The manuscript on the right-hand side comes from Novartis, and they carried out a prospective prediction of human pharmacokinetics for 18 compounds, and their conclusion was that their prospective human PK prediction methods yielded good prediction results. We then move on to two more publications. The one on the left-hand side, a novel strategy for physiologically based predictions of human pharmacokinetics carried out at Roche, concluded that in the majority of cases, PBPK gave more accurate predictions of pharmacokinetic parameters and plasma concentration time profiles than the Dedrick approach. And on the right-hand side, we have a manuscript from Johnson & Johnson, where they looked at prediction of human pharmacokinetics using physiologically based modeling. And this was a retrospective analysis of 26 clinically tested drugs. And their evaluation demonstrated that PBPK models can lead to reasonable predictions of human pharmacokinetics. In addition to these manuscripts, there have also been guidance from regulatory agencies for PBPK modeling. On the left, we see the physiologically based pharmacokinetic analyses, format and content guidance for industry from the FDA, and this came out in August of 2018. And on the right-hand side, we see the European Medicines Agency guideline on the reporting of physiologically based pharmacokinetic modeling and simulation, and this came out in 2019. So we've seen that PBPK is used to make first-in-human pharmacokinetic predictions. And we've also seen that there are a number of manuscripts highlighting the accuracy and the value of applying PBPK for first-in-human pharmacokinetic predictions. Now we will focus on the key components of a first-in-human PK prediction for oral administration. And those are the oral absorption. This is the multifactorial process driving drug reaching the systemic circulation. Once the drug reaches systemic circulation, it is then distributed. And here we will use, or you use, mechanistic tissue partitioning equations to describe the distribution, the partitioning of the compound into the tissues. And finally, we have metabolism and elimination. This is where you need a quantitative understanding of the main mechanisms of drug clearance. So let's look at those mechanisms in the form of a diagram. So here we have a 30,000 foot view for a first-in-human PK prediction for oral administration. And the model combines the advanced compartmental absorption and transit model, where we have those 9 compartments from the stomach down to the ascending colon. This model is combined with a systemic disposition model, which comprises of the tissues that make up the body. So the 13 tissues, which includes the liver, the muscle, the heart, the skin, the adipose, etc. If we now look at the ground level view of the PK model for a first-in-human prediction following oral administration, we will look at the individual ACAT compartments. And here we see transit in of the drug. This could be as unreleased, undissolved, or dissolved material into the lumen of the particular compartment that we're focusing on. The undissolved material may dissolve. That dissolved material could precipitate or could be degraded. Both the undissolved and the dissolved material could transit out of this compartment into the next GI tract compartment. The dissolved material, the dissolved drug, the dissolved compound could passively permeate, be absorbed into the enterocytes, or it could be actively absorbed or actively secreted back into the lumen. Once in the enterocytes, the compound could be metabolized in the gut wall, or it could be passively or actively transported into the systemic circulation. So there's many processes occurring in each compartment simultaneously in an ACAT model. If we now look at the diagrams at the bottom of this slide, we see two representations of the tissues within a systemic disposition PK model. The first is the perfusion limited tissue. This is the more simpler model where we have a vascular plasma compartment and the tissue space. The more complicated permeability limited tissue model has the vascular space, the extracellular space, and the intracellular space. And we can see passive transport of the compound from the extracellular to the intracellular, driven by the permeability surface area product, or we have the active transport of the compound with the Vmax and the Km. From these diagrams and focusing on the many mechanisms and processes that are occurring, I hope or I think you'll agree that a PBPK model for first-in-human prediction following oral administration is very complicated. And so what we need is a consistent strategy for building those models so that we can ensure the quality. And that strategy was provided to us by the Pharma GastroPlus user group. So they defined a first-in-human PBPK strategy for modeling following oral administration. And so there are flow diagrams for each essential component of a first-in-human prediction using PBPK modeling. This is an open access manuscript, and I believe at the time of me making this presentation that this publication has been cited 60 times, or at least 60 times, and has been accessed around 13,000 times. Let's look at those flow diagrams provided for us by the Pharma GastroPlus user group. The first flow diagram encourages you to combine QSPR with PBPK. QSPR is quantitative structure property relationship models. And if you combine that with PBPK, you can get a good understanding of the compound, and probably more importantly, you can identify those experiments which are likely to be critical to ensuring that you have or you build a quality PBPK model. So if we look at this flow diagram, the first question that you would ask is, what is the compound type that you're working with? Is it acidic? Is it basic? Is it amphoteric? Is it neutral? And then is that compound lipophilic or is it hydrophilic? Then what type of compound in terms of the biopharmaceutical classification system are you working with? Is it a high solubility, high permeability, or is it the more common high permeability, low solubility BCS class 2 compound? And then you can use the extended clearance classification system to determine the clearance mechanism. Is the compound predominantly or most likely to be cleared by metabolism, or is it likely to be cleared by renal elimination? And when you've carried out this assessment, you then have an understanding of the compound. So for example, if you're working with a lipophilic basic compound, which has poor solubility, you'd be looking to measure the solubility early because you're likely to see dissolution in the stomach and then precipitation as the compound moves into the duodenum. So you need to understand the solubility. And for the systemic distribution of a lipophilic basic compound, you would consider measuring the blood to plasma ratio earlier on, because that is likely to be a sensitive, a critical input into the KP equations, which describe the partitioning of the compound in the tissue. Once you have an understanding of the compound, you then focus on the individual mechanisms. So here we have a flow diagram for distribution. And the starting point, the question that you ask yourself is the passive distribution in preclinical species predicting using the Lukachova, the standard default equation in GastroPlus, with measured input data for lipophilicity, pKa's, plasma protein binding, and blood to plasma ratio? If your compound has low permeability and you think that it may be that the disposition is driven by the transporters, you would then need kinetic data for the compound. And you would know that by carrying out the in silico assessment that will guide you in your thinking. So if the passive distribution is predicted in the preclinical species using the Lukachova equation, so if we go down here and it's yes, you could then use that method to predict for humans. If the answer is no, then you would look to your project team to see if there are any tissue distribution or QWBA data available, more than likely in the rat, to help model the distribution of the compound in the rat. If those data are not available, and they don't tend to be routinely available, you would then be moving down the diagram here. And most of the time, you end up in the left-hand side, where you're adjusting the predicted KP values via one of the inputs. So for a basic compound, it may be via the blood to plasma ratio. And then you're looking to accurately predict with that adjustment in the preclinical species. And if you can, you would then use that adjustment for the human prediction. If this method doesn't work and you're working with a permeability limited compound, then you may well end up down this part of the flow diagram here, where for permeability, you would be looking to see whether a specific PSTC, the permeability surface area product, could predict in the preclinical species. And then if it does, you would use that value to predict for human. Moving on from distribution, we then focus on metabolism and elimination. So based on the preclinical data and the extended clearance classification system, you need to identify what is the contribution of each mechanism of elimination for your compound. And again, consider the impact of transporters if your compound is predicted or indeed is measured to have low permeability. So the mechanisms that we're focusing on are metabolism. So if your compound is predicted to be cleared predominantly by metabolism, can you establish an in vitro, in vivo extrapolation in the preclinical species that verifies that you can use microsomes or hepatocytes to predict the clearance in the preclinical species? And then you would use that liver fraction to predict the clearance in human. If your compound is predicted to be renally eliminated, the first thing that you would assess is, does the product of glomerular filtration and fraction unbound in plasma predict the renal clearance in the preclinical species? If it does, then you would use that method to predict for human. If it doesn't, then you would need more mechanistic models for renal elimination that potentially include secretion and reabsorption, and you would look to verify those in the preclinical species. And finally, if your compound is predicted to be biliary eliminated, then this is going to be more challenging to accurately predict in human, but it is possible. You could use systems such as sandwich cultured hepatocytes and mechanistic in vitro transport data to predict the biliary secretion in the preclinical species and then translate to human. If none of these mechanisms can be accurately verified within the preclinical species, you may have to consider using empirical scaling factors to predict the clearance in humans using that preclinical data. Moving on from metabolism and elimination, we now focus on oral absorption. And what we're asking here is absorption in preclinical species predicted using measured solubility and in vitro permeability data with an ACAT model, with the Advanced Compartmental Absorption Transit model? When you're modeling oral absorption data in the preclinical species, you're more than likely going to have IV data in those species. And if you do, it is a good idea to fit a compartmental PK model so that the systemic disposition is accurately modeled in that preclinical species. You should at this point consider the effect of formulation of food on oral absorption, and the solubility data that you use must be for the same form of the compound that was dosed. You would also look to establish a correlation for the in vitro permeability assay with in vivo permeability using clinical standards. And so if the absorption is predicted in the preclinical species using the measured solubility and permeability data, you would then apply that methodology to predict for human. If it doesn't predict, then you would need to perform a parameter sensitivity analysis to determine the most sensitive input. So is the oral absorption driven by solubility and dissolution? If it is, then can the bile salt solubilization ratio be estimated from biorelevant solubilities and predict for the preclinical species? If it can, you would use that data to predict for human. If not, if you assess the variability, the known variability on the volumes in the lumen of the GI tract, if you have to adjust those to predict, it's not recommended to change physiological parameters, but the volume in the GI tract is known to be uncertain. Therefore, you may qualify or verify percentages in the preclinical species that you can apply to human. If dissolution is poorly predicted, then you'll be looking to see, can measured particle size data or dissolution data help accurately predict in the preclinical species? If they can, you would use those data for human. If your parameter sensitivity analysis determined that permeability was driving the oral absorption, then you would be looking at the permeability value itself, or potentially the ASF model, the absorption scale factor model, to see how that might be driving the prediction, and then translate any findings to human for the first in human prediction. And finally, if other processes are driving that oral absorption, you are more than likely going to need additional quantitative data to describe that process accurately in your PBPK model. And now we look at the final flow diagram that focuses on one of the mechanistic components of a PBPK model following oral administration. And this is the flow diagram that focuses on gut wall metabolism. And here we're asking, is the compound metabolized by enzymes which are known to be active in the gut wall? If it isn't, then significant leverage, we look to use the in vitro data that we have describing the hepatic liver metabolism to make an estimate of the gut wall metabolism. So we'd ask ourselves, are the enzymes expressed in the liver? If they are, do we know the contribution of the individual enzymes to the hepatic metabolism? If we don't, then we'd have to make an assumption that all of the metabolism is via the enzyme with the highest abundance in the gut wall, and then scale from liver microsomes or hepatocytes to the gut wall. And this is likely to result in an under-prediction of FG and over-prediction of the metabolism. If we do know the contribution of the individual enzymes, then you would use the relative expression between the gut and the liver if that's known. If you do have Vmax and Km data, then you can use those to predict for the specific metabolism in the gut wall. If you don't, you'd use the intrinsic clearance and assume non-saturable metabolism in the gut wall. And this again is likely to result in an over-prediction of the metabolism in the gut wall. The final flow diagram in the manuscript from the PharmaGastro Plus user group doesn't focus on a mechanism. It highlights the importance of always including uncertainty in your first in human PK predictions using PBPK. At first glance, this flow diagram can seem very complicated and a little bit overpowering. But in reality, for your specific compound, there will only be a couple of parameters that are sensitive and should be explored in terms of uncertainty. And so this flow diagram is essentially a checklist for you to go through and help identify which two parameters are most likely to have the biggest impact, to be the most uncertain. And it's those parameters that you would combine in a 3D PSA. So for a compound that's lipophilic with poor solubility and is basic in nature, you would be looking at combining perhaps the reference solubility in the stomach with the blood to plasma ratio, which is going to drive the systemic disposition, as two parameters which may dictate the level of uncertainty in your prediction. So we've discussed what PBPK is and what PBPK isn't. We've looked at the application of PBPK for first-in-human predictions. Now I'd like to come to some case studies for first-in-human PBPK modeling. Now, there are 4 case studies in the Pharma Gastro Plus user group that you can read or have a look at. But here I want to focus on a couple of case studies from different manuscripts. So first, we'll look at Kisqali. And this is where PBPK modeling had an impact on the drug label. So this is a CDK inhibitor improved for first-line treatment of breast cancer. The compound is a weak base with a pH-dependent solubility profile over the range found in the human gastrointestinal tract. Now, many cancer patients are treated with acid-reducing agents that elevate the gastric pH. So therefore, there's a potential interaction between acid-reducing agents and Kisqali because of this pH-dependent solubility of the compound. And here is the publication by Samant et al. From 2018 that gives you the detail behind this case study. So in this example, they didn't combine the ACAP model with a PBPK model for systemic disposition. They combined the ACAP model with a compartmental PK model. So it's a 2-compartment model where you have a central compartment and then you have distribution to and from that peripheral compartment. So the model was fitted using PK data from healthy volunteers. And then the model was subsequently qualified using PK data from additional healthy volunteer studies and also data in patients. From this figure here, you can see that the model adequately simulated the PK profile of the compound in fasted, healthy subjects following a dose of 600 mg. We have the simulated plasma concentrations with the solid line, and we have the observed data represented by squares. The model was then qualified using PK profiles from patients with cancer in this subsequent study. And again, you can see the good prediction where the simulated plasma concentration, the solid line, passes very closely to the observed data, which is represented by the squares in the plot. So the verified model was then applied to carry out a sensitivity analysis examining the effects of varying the gastric pH in the range of 1.5 to 8. And they had a look at the effect on the absorption. And from the plot here, you can see that the fraction absorbed remained consistent over the pH range from 0.5 up to 8. So the outcome of the PBPK modeling, which was used alongside POP-PK and NCA in a multi-pronged approach, was that a dedicated PPI, proton pump inhibitor trial, was not required. And the modeling, as you can see from the bottom here, was actually used to support the labeling language. So co-administration of ribociclib with drugs that elevate the gastric pH was not evaluated in a clinical trial. However, altered absorption was not identified in POP-PK and was not predicted using physiologically based PK models. So the modeling, this multi-pronged approach, avoided a dedicated clinical trial. The next case study that we will discuss comes from a publication by Tycho Heimbach et al. In 2009. And here they highlight that accurate predictions of human pharmacokinetic and pharmacodynamic profiles are critical in early drug development. That is, they are critical for formulation risk identification, lead candidate differentiation, and prediction of clinical outcome. And the manuscript covered practical projection approaches for formulation-dependent human PK/PD parameters and profiles of BCS classes 1 to 4 based on preclinical data. There are multiple case studies in this publication, but there was only one that truly used PBPK. So this is the example I'm focusing on here. And they used the PBPK for predicting the distribution using GastroPlus. They combined that with allometric scaling for the clearance, because as you saw when we went through the metabolism and elimination flow diagram, sometimes you do have to use empirical scaling methods for clearance. But they highlighted, and this was say back in 2009, that they were predicting profiles, not just parameters, because at that time there was a big focus on parameters and people weren't routinely predicting profiles. Also, back then, these type of predictions did not always factor in biopharmaceutics, but this example in this manuscript does. And if you look on the right-hand side of this slide, you'll see the schematic, the flow diagram of the approach that they applied for this particular example. So the compound that we're focusing on from the manuscript is compound R. And this was or is a novel antiviral drug. So the primary target was to use this in an intravenous dosing hospital setting. But for community use of the compound, oral dosage forms were also going to be developed. So the human PK modeling was performed with the following goals. They wanted to predict the intravenous PK profile following a 75 mg infusion. They then wanted to determine if compound R could be dosed orally and achieve a bioavailability greater than 50%. And finally, they wanted to predict whether food effects would impact the PK parameters. So they carried out their modeling using GastroPlus, and they used the Leukotrophia default KP method with measured pKa and log P as inputs. And this approach adequately predicted the pharmacokinetics in the rat following IV dosing, as you can see from the figure here. They then used this approach to simulate the IV profile in human, and this also gave an adequate prediction, as you can see in the plot of the figure on the right-hand side. Moving on to the oral PBPK predictions, they again used GastroPlus, and they used the default physiology model for a fasted human. And this simulation of the oral profile after a 75 mg oral SED solution administration was adequate when they got the clinical data. And you can see from the figure down here. Unfortunately, for the prediction of food effects, there was a large prediction error, and this was thought to be due to the true in vivo human solubility being unknown for this compound. The outcome for this case study is that the use of integrated softwares such as GastroPlus in combination with established PK prediction methods allows the projection of formulation-dependent preclinical and human PK/PD profiles required for compound differentiation and development risk assessments. So we come back to the take-home messages that we discussed at the beginning of this presentation. Hopefully, you'll agree that we've seen an overview of what PBPK is and how it is used. The standard definition of PBPK is a mathematical modeling technique for predicting the absorption, distribution, metabolism, and excretion, the ADME, of compounds in humans and animals using physiology and compound-specific physical-chemical data. We've seen with the publication by the FDA that PBPK is used for many applications. We then focused on the application of PBPK for first-in-human predictions. And I would like to highlight here that you really need to utilize the power of preclinical verification. Can you adequately and accurately predict the PK profiles in the preclinical species? And if you can, you've got the most confidence you can have when you apply PBPK for first-in-human predictions. And finally, we looked at a couple of case studies. The first one highlighted how modeling supported the drug label language without dedicated clinical interaction studies. And then we looked at the anticipation of human efficacious doses with a publication which is over 10 years old now. And I do believe that we will be seeing more examples in the future of this application of PBPK for human efficacious doses. The reason I believe that we'll see more examples of anticipation of human efficacious doses using PBPK is that we're starting to see them now. So this publication came out in February of this year, and this publication was from GSK, talking about their compound 2556286. So here, human PK parameters were calculated for the compound using PBPK. These PBPK models accurately predicted the IV and oral PK data from preclinical species. So they're doing the preclinical verification. And then they combined this model with some PD data to predict a dose in humans. So I think this is one example that is just come out and we'll be seeing many more soon. So that is the end of the presentation. And if you would like more information from us, then please visit our website at www.simulations-plus.com, or you can email us at either of these email addresses, one for training and one for general inquiries. I'll now move on to answering some questions that were supplied to me prior to the presentation today. Now we come to the question and answer section of this presentation. So question number one. Currently, many pharmaceutical companies use PBPK modeling to predict first-in-human PK and submit them to the FDA and EMA. What are the main purposes and applications of these PBPK models? Well, if we refer back to the presentation, we will remember that according to the literature, the main current application of PBPK models submitted to the FDA is drug-drug interactions. And the aim would be to avoid a clinical DDI study. We also saw that the application of PBPK for first-in-human is increasing, and the aim is to show that the PK of the compound is well understood in animals prior to dosing in humans. And with that last example I showed you for GSK, you saw how they combined PBPK with PD data to estimate the human efficacious dose. We now move on to question 2. What is the FDA and EMA's acceptance of the use of PBPK models to predict first-in-human PK? And are there any case studies that could be shared in the IND submission? So I do not have statistics on the acceptance of PBPK models for first-in-human by regulatory agencies. But as we've seen, there are examples in the literature of successful case studies. For example, Kisqali and the labeling language without the dedicated PPI trial. So question 3 asks, can you share some specific cases where PBPK was used in first-in-human PK research and accelerated clinical research progress? So as we've referred to a couple of times, we have the Kisqali example and the labeling language, which meant that a dedicated PPI trial was not required, and therefore it accelerated the clinical research or progression of this compound. Another example would be from the Pharma Gastro Plus user group publication. In particular, compound 1 in this paper highlighted how the extrapolation of pharmacokinetics was required to progress, to estimate a clinical dose for the compound and progress this compound. And the modeling highlighted that a food effect was likely to occur with the compound. So the first clinical study was indeed conducted in the fed state based on the modeling. So this was a nice example of how there was reason to believe in the compound based on the PBPK modeling, and the PBPK modeling also dictated how the first clinical study was conducted. The next question, question 4. When we extrapolate human pharmacokinetics using different PBPK models in preclinical species, how do we select suitable animals PK to make the prediction more accurate if the results are quite different using different animals? Well, the answer here is that all animals are suitable if their PK is understood, and that you have the right input data, the right in vitro data generated to parameterize the mechanisms that are important in each of those species. Now, if one or more of those preclinical species are not predicted well, then this does highlight that there may be a missing mechanism. And in reality, the ultimate species for human is human. As long as you understand all of the animal data, you have the highest confidence that you can accurately predict for human. But in reality, we don't know until you get into the clinic. Question 5. If animals are shown the nonlinear PK, or if animals are exhibiting nonlinear PK, what are the key points for using PBPK prediction? How do we ensure the prediction accuracy? Well, what you need to do is identify the mechanism behind the nonlinearity in the pharmacokinetics, and then measure the appropriate input parameters to characterize that mechanism. For example, is the mechanism absorption, distribution, or elimination? So if it's absorption and there's nonlinearity around the transporters, you would need to measure data that would be able to accurately parameterize that nonlinearity in absorption in the transporters in the preclinical species. Question number 6. How do we evaluate the accuracy of a PBPK prediction without clinical PK data? So the answer to this is, the key is preclinical verification. Can the pharmacokinetics be accurately predicted in the species, in the animal species for which PK data are available? If so, then you have as much confidence as is possible before going into the clinic. But as I said, there are no guarantees that you will accurately predict in human, but you're giving yourself the greatest chance of success if the PK data in the preclinical species can be verified. Question number 7. Could you give an example on how to guide early formulation development after completing first-in-human PK prediction using PBPK models? So the example I will give here is the case study from the Tycho Heimbach et al. Publication in 2009. So we looked at compound R, for which the primary target was intravenous dosing in a hospital setting. However, for community use, oral dosage forms were also to be developed. So they carried out the first-in-human PK predictions following a 75 mg intravenous infusion dose. And then for the formulation development, they made predictions following oral dosing to assess whether they could achieve a bioavailability greater than 50%. Question number 8. What is the applicability of PBPK, NO-AEL, MABEL, and allometric scaling to predict the starting dose of first-in-human? Do these methods require cross-validation? If the results predicted by these 4 methods are very different, How do you determine which result is more reasonable? Well, in my opinion, currently PBPK can't be used alone to predict the starting dose for first in human, and the predictions need to be compared with the other established, more empirical methods. If the results are different, then you do need to take the lowest safety dose into the clinic. Hopefully, in time, we will see PBPK modeling be able to be used alone to predict the starting dose, but we're not there yet. Question number 9. Can PBPK use to predict first-in-human PK for bio-macromolecules such as ADC and antibody? What are the advantages and disadvantages? What is the main difference from small molecule prediction of PK? So yes, PBPK can be used for large molecules, as was mentioned in the presentation, and it is more mechanistic than empirical approaches. But I think it's fair to say that currently there is more experience of and functionality for modeling small molecules using commercial platforms. But I do expect commercial PBPK platforms to be used more routinely in the future for predicting large molecules. Question number 10. Can first-in-human PK predictions be used to predict other drug delivery routes? And for inhalation, what data are needed for animal PBPK models? Well, the answer is yes. PBPK can be used to predict first-in-human PK for other delivery routes than oral. For inhalation administration, ideally you would have the following data. So you would have the standard in vitro data for PBPK models, so lipophilicity, solubility, plasma protein binding, etc. You would also have intravenous PK data in animals to help characterize the systemic disposition. You would have oral PK data in animals to characterize any swallowed portion of the inhaled dose. You would also have inhaled PK data in animals to understand the pulmonary absorption. In addition, you would ideally have route-specific data such as pulmonary solubility, pulmonary permeability, and deposition following inhalation.
So that's okay. 非常感谢今天晚上 Nell 的介绍。今天的话 Nell 的话主要讲了什么是 PBPK 以及 PBPK 模型的应用，以及介绍了采用 PBPK 模型预测首次人体 PK 的策略，包括分布、代谢、吸收、肠壁代谢的考虑等。最后的话，通过2个案例，第一个的话是通过 PBPK 模型减少 PPI 临床试验的案例，第二个的话是如何预测人体的有效剂量，并且的话，针对我们事先征集的10个问题，给大家做了一一的解答。然后下面的话是我们进入本次公开课的问题解答的第2个部分。然后我这边的话已经将问题发给了 Nell， 然后下面的话有请 Nell 来回答今天晚上第2部分的这个问题。Hi Nell, can you hear me?
Yes, I can.
You can answer the question about I sent to you by Teams.
Sure. Hello, everyone. It's nice to meet you virtually. So the first question is, PBPK modeling requires a lot of parameters and more understanding of the mechanism. How should companies design PBPK studies when modeling data is insufficient? So yes, PBPK modeling does require a lot of parameters, but most of those can be predicted from structure. Certainly for simple models. For more complicated models with transporters, you will need data. But for most models, you can predict a lot of the parameters from structure. The second part of the question, how should companies design? I think that should be design their DMPK studies. Those studies should be designed with the end in mind. For example, if you want to assess the exposure of the compound in a specific tissue in a preclinical species, for example, the muscle, you would look to sample the muscle, design the study to sample the muscle with the appropriate sampling time points to define that concentration curve. And you can use the modeling to guide that. So you should always design your DMPK studies with the end in mind. And then the modeling can be used to understand that data. And if there's anything missing, if the input data is insufficient to predict the in vivo outcome, you would then look deeper into the mechanisms, deeper into the compound to see what parameters are missing. If I continue with the example of modeling in the muscle, if we have a basic compound and we're not predicting the partitioning into the muscle, it may be that we don't have an accurate value for the blood to plasma ratio. If that is the case, we would then design and run an in vitro study. So to summarize, PBPK modeling does need many parameters and includes lots of mechanisms. The parameters can be predicted from structure, and your DMPK, your in vivo studies, should be designed with the end in mind, and modeling can be used to inform and understand the data that you get. If I now look at the second question, the second question says, in the IND stage, the compound is a transporter substrate. How to use the PBPK model for human prediction? So if you have a compound which is a substrate for a transporter, you will need the transporter kinetic data to include that mechanism in your model. So you will need the Vmax and the Km. They can be measured from in vitro systems, and you can then use those inputs to predict for in vivo. They may need to be refined, but we do have examples where in vitro data are predictive of the in vivo situation. So to summarize, if your compound is a substrate for a transporter, to include that in the model, you will need transporter kinetics. You can explore before you measure the transporter kinetics with the modeling to see what values you may expect, but you would need to measure those in vitro values to inform your model. The next question we have is controlled release dosage forms. Good controlled release was achieved in dogs and monkeys, but not in humans. Can such formulations be predicted by the PBPK model? And what are the possible differences between animals and human? The answer is yes, PBPK models can be used to predict such formulations. But what we would need to know is, was the controlled release dosage form the same in the animals and in humans? And if it was, we would need one set of data to describe that controlled release. And then we combine that with the physiology to predict for the individual species. So if the human wasn't predicted well, it may be because the controlled release form wasn't characterized well, or the physiology in the model was not appropriate. So you could explore both scenarios with the model. With modeling, you can carry out a what if scenario to explore the range of parameters that predict the data. Those values need to be plausible, but they can inform your modeling and your understanding of the compound. So yes, PBPK modeling can be used to predict following controlled release. It can be used to understand differences in preclinical species and humans, but we need good quality input data to describe the formulation, the release of the drug from the formulation, and also the appropriate physiology. The next question is, does the PBPK model have any applications in generic drugs? Yes, it does. A PBPK model can help you understand the generic drug and may help you get that generic drug accepted quicker? That's a very timely question, because at Simulations Plus, we are in the process of developing a GastroPlus database with compounds that are relevant to generics. And with those characterized models, they can be applied to facilitate, to speed up the acceptance of those generic drugs. So if you'd like more information about the what we are calling the Biopharmaceutics Task Squad, then please send us an email. At the end of the slides, there are emails here. So if you email us at info@simulationsplus.com, we can give you more information about the generics drug database that we are currently building. I think that's all the questions I see for the moment in the chat. Are there any more questions? Ethan, I can't hear you, Ethan, if you're talking. Ethan, I can't hear you. I think you're on mute.
Daniel, can you share your
Yes.
experience about the novel? Okay, I have sent the question by Teams.
Yep. Thank you. Okay, can you share your experience about the novices, how to learn and improve their knowledge and skill on first-in-human PK prediction using PBPK? Firstly, I would say I think we are all novices. We are always learning, and with every opportunity, there is more to learn. When you're new to this, obviously there is more to learn, but as I say, we're always learning. And what I would suggest is number one is to read relevant manuscripts. So, for example, when manuscripts like the one on the screen come out, read those to learn from what other companies are doing in this space. So that's number one, read publications. Number two would be to attend any relevant training courses. We ran a first-in-human applications-based training course earlier this year at Simulations Plus. I'm sure there are others out there. So I would suggest attending training courses that are relevant. And the third thing is, and probably the most important thing, is to practice, is to apply PBPK modeling first in human to your compounds, because that is where you learn the most. You learn where your gaps are, and then when you know where the gaps are, you can obviously look to fill those. So I would summarize, we are all learning always, and to learn, I would read, I would attend training courses, and I would apply to my compounds or your compounds.
Can you share a few PBK books?
A PBPK book.
Yes.
There is one book, and I think I can share. So here we've got physiologically based. If I move that up, move it into here. So this book is by Sheila Annie Peters. It's physiologically based pharmacokinetic modeling and simulations, and I believe she's brought out a new edition. So I'll hold that up again.
Okay, I have this book, and I will send it to the participants. Thank you, Neil. I have no more questions. Thank you for your share about PBK in first home.
Thank you for the opportunity. Nice to meet everybody virtually, and use the email address if you have any questions, because I will learn from your questions. So contact us if you'd like any more help.
Okay, thank you.
Thank you. Have a great evening, everyone.